This is our third and final lecture on fire blight. In the second lecture on fire blight, we learned that Erwinia ameliivora can be present on tissue surfaces without causing disease, and that this is known as the epiphytic phase of the pathogen. We learned that these epiphytic bacteria are spread by insects and rain or heavy dew, that the optimal temperature range for bacterial growth and infection is 70 to 80 degrees Fahrenheit, and that infections occur through the flower nectaries. Lastly, the disease cycle was presented, and we learned the source of primary and secondary inoculum. In this lecture, we will focus on how to manage fire blight, considering each of the phases of the disease. The learning outcomes for this lecture are: number 1, identify and describe disease management strategies for fire blight of apple and pear, and 2, using your knowledge of the pathogen and disease cycle, develop a disease management program. As with all diseases, an integrated disease management approach that considers host resistance, cultural practices, and sanitation practices, disease forecasting systems, and chemicals is recommended. Because fire blight is a systemic disease, strategies that prevent the introduction of the pathogen into the orchard are very important. But because the pathogen has an epiphytic phase, strategies that prevent the spread of the bacteria within the tree and from tree to tree are also very important. There are a few rootstocks with resistance to fire blight, and breeding programs make fire blight a priority because an infection in the rootstock can kill a tree very quickly. The older rootstocks, including Bud 9 and M9 and M26, are very susceptible to fire blight. However, the newer rootstock varieties, including those in the Geneva series, as well as M7, are resistant to fire blight. And many of the new plantings that are being done in the Northeast are using the Geneva rootstocks or M7. Resistance in cultivars that are favored by the consumer is minimal. And as you can see here, the most popular varieties by consumers, such as Fuji, Galley, and Honeycrisp, are very susceptible or moderately susceptible to fire blight. The level of reported resistance, however, is not very reliable. So for example, Golden Delicious, which is reportedly moderately susceptible, as shown here. However, when it's compared side by side with Jonathan, which is very susceptible, in a year where the conditions are favorable for disease, Golden Delicious, shown here on the right, actually has a resistant phenotype when it's compared right next to Jonathan, which is very susceptible. There are a few rootstock options for pear as well, although fewer than those available for apple, and only a couple of them are resistant. So there are 2 rootstocks with resistance to fire blight. Those include Old Home and Old Home crossed with Farmingdale. Similar to apple, most of the popular pear varieties are susceptible to fire blight. As shown here, Bartlett, or Aurora, and Bosque, which are the 3 most popular varieties with consumers, they are all very susceptible to fire blight. Producers will need to implement a stringent management program for controlling fire blight of pear because, as you can see here, there are not a lot of varieties that are resistant. In addition, the growers will often isolate their pear plantings from their apple plantings to try and quarantine the pathogen and prevent cross-contamination to their apple plantings. So if we start with the holdover canker phase or the canker phase of fire blight, it's critical that this phase is managed during the dormant season before the cankers begin to ooze. So dormant season pruning will reduce primary inoculum in the spring. So limbs with cankers should be cut out and the debris removed from the orchard and burned. The first cut should be made about 4 inches below the canker. So if you look here on the right, you can see a visible canker. So the grower should go ahead and make a cut 4 inches below that. And then they should continue trimming the tree or the limb until healthy tissue is observed. Holdover cankers can also be treated. If they're going to be treated, it needs to be done in the dormant season prior to when the trees are at the green tip stage. And that's because copper applications are about the only chemical that are effective at killing the bacteria at this stage. So copper applications will again reduce the primary inoculum in the spring. Some of the formulations, however, are phytotoxic to green tissue, and that is why it's important that the copper is applied before green tip. However, some of the newer formulations of copper are more compatible with green apple tissue and can be used past green tip. So it's important for growers to read the label to determine whether or not there might be phytotoxicity to the green tissue on their apples. Application of insecticides or horticultural oils at this time are also important because they will prevent insects from spreading the bacteria that's oozing from the canker. So the insects will come in contact with the ooze and then they can spread it to the flowers. There are 2 models that can be used to predict infection risk levels during the bloom season. And these models are used in order to determine when's the best timing to apply antibiotics to control the blossom blight stage of the disease. So there are 2 models available, cougar blight and Mary blight. And both of these models consider 4 main variables. The first variable is first bloom date. They also consider fire blight history in the orchard, which is considering the presence or absence of inoculum in the orchard or in nearby orchards. And then the third one is temperatures, and the temperatures that are used to predict when epiphytic populations are at levels that would cause infection. And if you remember from our previous lecture, that would be levels of 10 to the 4 cells or higher. And then blossom wetting period is also considered. And we'll go over these 4 in a little bit more detail. So for fire blight history, the models give a score of 1, 2, or 3, depending on the history of fire blight in the orchard. So a score of 1 is given if there was fire blight occurring in the orchard in the previous year and it is active this year. And in order to determine if it's active, the growers will need to go out and scout their trees and look for cankers or any other tissue that might be oozing. A score of 2 is given if fire blight occurred in the previous year, but it has not been observed this year. And a score of 3 is given if it did not occur last year and has not been seen this year. So a score of 1 has more weight in predicting risk than a score of 3. So temperature is input into the model as a temperature risk value, or TRV, or an epiphytic infection potential value, or EIP. And that and the value that is used depends on the model. So these values relate to the average hourly temperature to population growth of the bacteria on the stigma. And so if you look over at the graph on the right, and again, this, you've seen this graph before, up at the peak here, is where you have your epiphytic infection potential at the highest. And these values are calculated over a 4-day period, or 96 hours. So blossom wetting period is also considered, with 2 hours or more of a wetting event after the epiphytic infection potential is required for infection. So they consider the EIP, and then if there are 2 hours or more of wetting, then the risk for infection is high. And they determine blossom wetness either using leaf wetness sensors or by collecting rain data using weather stations. In addition to the 4 variables I just mentioned, average daily temperature is also considered. And so if temperatures are 60 degrees or above, then that gets considered in the model in order to predict risk level. And these programs generate prediction maps or tables that can be used by the grower. The grower will review these prediction maps or tables and then determine whether or not they need to apply an antibiotic. So this output that I'm showing here is from the Network for Environment and Weather Applications, also called NUA, which is based out of Cornell University. And the outputs can vary depending on the organization providing the software. For example, the Ontario Ministry of Agriculture, Food and Rural Affairs provides a prediction map for the tree fruit production regions in Ontario. So with the NUA output, as I said, it's a table, and you can very clearly see with this table that it's considering the 4 variables that I just described. So the history of fire blight, the grower can select that their history, and then that will input a score of 1 or 2 or 3 into the model. First blossom date can be entered, or they can leave that blank and it will default to when Macintosh apples first bloom. And so not all varieties bloom at the same time. And so incorporating the first bloom date can be important, because if you're basing it only on Macintosh, you may actually miss the actual bloom date for the variety that you have planted. Temperature again is considered, and this is the infection potential value. And then the wetting period. And the wetting period is calculated based on the amount of rain, whether or not there's a presence or absence of dew, and it also can consider leaf wetness. So based on the risk level, the growers will decide whether or not they will apply antibiotics. And risk is shown as different colors, which makes it easy for the grower. So if you have a green color, your risk of an infection occurring is low. If you have a yellow color, then it is moderate. Orange means that you have a high risk of an infection, and then red means that an infection will occur. Okay, so currently antibiotics are the only form of control for blossom and spur blight. And antibiotics are most effective when they're applied within 24 hours before an infection event, and are also effective within 24 hours after an infection occurs. However, the efficacy of the antibiotic drops each hour after an infection event. Full coverage of the interior of the flowers is essential for control to occur. And this is because antibiotics are a contact bactericide, so they must come in contact with the pathogen, and the pathogen is located at highest numbers on the stigma of the plant. So right here in the center. So if the flowers are closed, as you can see, some of them are in this image, then the antibiotic is not going to reach the bacteria and won't be effective. So multiple sprays are needed because of this, and bloom, depending on the variety, can last anywhere from 1 to 3 weeks. And not only does that depend on the variety, but it also depends on temperatures. So antibiotics do not control the shoot disease phase, and therefore they can only be used at the blossom and spur blight phase. So antibiotics are prohibited for organic production, and therefore it makes it very difficult for organic apple producers to control the blossom and spur blight phase of fire blight, and therefore they really need to focus on the holdover cankers or the shoot blight phase. So this table here shows the 3 antibiotics that are registered for use in Ohio, and those include streptomycin, kasugamycin, and oxytetracycline. And the efficacy of these antibiotics varies somewhat, with streptomycin being the most effective and oxytetracycline the least effective. However, this is only true if resistant Erwinia are not present in the orchard. So most growers use streptomycin because it's inexpensive compared to kasugamycin and more effective than oxytetracycline. If there is streptomycin resistance in the orchard, then the growers generally switch to oxytetracycline because it is very inexpensive, even though it's less effective than kasugamycin. But kasugamycin right now isn't very cost effective for growers, especially considering the number of applications that have to be applied during bloom. The shoot blight phase of fire blight is very difficult to control, and that's because antibiotics are ineffective at this phase and because copper is phytotoxic. So in Ohio, trees will put on new shoots until early July, and then they will set a terminal bud after that. So applying fertilizer, specifically nitrogen, during this period can promote rapid shoot growth. And because young shoots and suckers are very susceptible to infections, the risk of disease increases. So maintaining a balanced fertility program is important because these new shoots are so susceptible. So fertilizer applications are recommended in the late fall or the very early spring. In seasons where weather favors an extended shoot growth period and disease pressure is high, an additional application of a growth regulator may be needed. We also recommend that growers only apply nitrogen based on soil and tissue tests, and that they conduct these tests annually. So again, just to reiterate that antibiotics are not effective and should not be applied to the shoot phase. So really the only option then for growers, if they want to manage new shoot infections, is summer pruning. And summer pruning can be effective in reducing secondary inoculum if it's done carefully. So we recommend that shoots be removed if they show symptoms, not signs, of fire blight. So if you see the wilting and the necrosis occurring, then summer pruning may be necessary. If it's done carefully and when environmental conditions are not favorable for ooze, splashing, or dispersal, it can be very effective. So when summer pruning is recommended, we recommend that they prune when the weather is dry to prevent splashing of any potential ooze that's present in the tree, that they use sharp and clean pruners so they get a very clean cut, that they cut into healthy tissue that's 12 to 15 inches below dead wood. And this is significantly higher than the dormant wood, but this is because at this time the bacteria are active and can be moving through the tree. Sanitation is recommended after every cut, and this can be done using 10% bleach solution. Unfortunately, this is very labor intensive, so most growers do not sanitize after every cut. Rather, they may sanitize after every tree or after every 2 or 3 trees. And lastly, similar to with dormant season pruning, it's important to burn or take the dead wood out of the orchard and put it in the trash. Lastly, I may not have mentioned this, but it's really critical that they do not prune if the trees are oozing. Okay, so because the shoot tissue is very susceptible to infections, growth regulators have been tested to determine if slowing the growth and reducing the amount of vegetative growth could reduce fire blight disease incidence and severity. And one specific growth regulator that was identified by George Sundin at Michigan State University was prohexadione calcium, which is marketed as a POGI, and now there's a generic version of it called Kudos. So POGI will, what it does is it inhibits gibberellin biosynthesis. And what this does then is it reduces the stimulation of stem elongation if it's applied to the trees after flowering. And additional studies have also shown that the growth regulator had no direct effect on the bacterium. But in addition to shortening the internodes, cortical parenchymal cell wall width nearly doubled in size. And this indirect effect of the doubling in size of the cortical parenchymal cells is referred to as the apogee effect. And if you look at the right here, I'm showing there are 2 shoots here that have been treated with the growth regulator, and you can see that they're significantly smaller than the one in the center that wasn't treated with the plant growth regulator. So these SEM images show the apogee effect. Non-treated shoots that are shown on the left here have thinner cortical parenchymal cell walls compared to those shown on the treated tissue on the right. So if you look at the cell wall structure here, you can see that it's much thinner than those that are shown in image B here on the right. And so these apogee treated cells have cells are thickening. The treated tissue also has thickened down here in an undefined way. And this effect that you're seeing here, both this undefined thickening as well as the cortical parenchymal cell thickening, is most apparent on young shoots. And as the shoots age, there is less of an effect. So this table shows the actual data from the study that was done in the Sundin lab. So the cell widths were measured on 3 ages of leaves. So leaf 1, leaf 2, and leaf 7. These are all different ages of leaves, and at 2 different time points. So they were measured 7 days post-treatment and then 13 days post-treatment. And they did these measurements both on non-treated and treated shoots. And if we look at leaf 1 and day 7 in this box here, you can see that the cell width nearly doubled in size. However, on the older leaves at day 7, so we'll go down to leaf 7 here, you can see that no effect was observed. And likewise, no effect was observed 13 days post-treatment in any of the leaves that were evaluated. So this indicates that the response is short-lived and that additional applications of the product might be needed. And in fact, the recommendations for using Apogee is that you put on one application and then 14 days later, you put on a second application. What I find really interesting is that the increase in cell width after one application was sufficient to build a cell wall barrier that doesn't allow the needle of the type 3 secretion system of Erwinia to pass the plant cell wall. So if you look at the image on the right, this is an image of a type 3 secretion system. And this system has a needle on it that's 2 micrometers in length. And this needle, what it does is it passes the host cell membrane and injects effectors into the host cells, and these injectors help to overcome the plant's host defense system. However, if the cell wall and membrane has thickened, this needle can actually not pass it. So you see that the 2 micrometers is similar to what the cell walls actually will thicken up to at 2 to 3 micrometers. So this is very, very interesting to me because it has an effect on the mechanisms that Erwinia uses to infect the plant tissue. In 2002, Dr. Mike Ellis did a trial in order to look at the effect of Apogee on both mean shoot length and the number of blighted shoots in a tree. And so he evaluated streptomycin, Serenade, streptomycin plus Apogee, and then a non-treated control. And if you're interested in learning more about this study, it was published as a plant disease management report by APS in 2002. So if we look at streptomycin and Apogee, you can see that the mean shoot length was significantly reduced compared to streptomycin alone, to Serenade, as well as to the non-treated control. So that's what we would expect with an application of Apogee. What was very interesting and really good to see was that the streptomycin plus Apogee also significantly reduced the number of blighted shoots per tree compared to all of the other treatments, as well as the non-treated control. And you can see that that reduction is very significant because this was a high disease pressure year where there was an average of 258 blighted shoots per tree. So the current recommendation to growers is that they use streptomycin at bloom, and then following bloom, they make 2 applications of Apogee. And growers in Ohio and surrounding states have seen really good control using both streptomycin and Apogee. So just to conclude, over the last decade, we have seen an increase in fire blight incidents, and there are several factors that could be contributing to this increase. The first is most likely due to the fact that more popular varieties are being planted, and that these more popular varieties are highly susceptible to fire blight. So these are your Honeycrisp, your Gala, and your Fuji varieties. We're also finding that growers are doing more of their own on-farm grafting. And on the right here, you can see a picture where they're grafting popular varieties, such as Honeycrisp, onto old trees or old rootstocks. And in doing so, they have the potential to introduce the pathogen into the system or into the tree. So unfortunately, grafting is a common mode of transmission for Erwinia, as well as other pathogens. And unless intensive sanitation practices are used, there is a high risk for introducing the pathogen into the system. And these intensive sanitation practices are really difficult to do in the field. Most grafting is done in a clean work area within a laboratory. So warmer winters may also be contributing to increased incidence as well as severity. So warmer winters and springs mean that holdover cankers may ooze earlier, trees may also bloom earlier, and this makes it difficult to adequately time applications or to apply applications of copper to the holdover cankers or streptomycin to the blooms. Remembering that copper is phytotoxic to green tissue, and therefore the window of time that copper can be applied might be shortened when we have warmer winters. Additionally, high-density plantings are more common, and in these high-density plantings, trees are planted anywhere from 2 to 5 feet apart, and therefore the dispersal distance of the pathogen is reduced compared to typical or non-high-density plantings where the trees are anywhere from 5 to 10 feet apart. And lastly, is that Erwinia amylivora populations are becoming resistant to streptomycin, and because of this, we will see an increase in disease, not only in incidence, but also in disease severity. And for the first time in Ohio this year, we have found that we do have several orchards with streptomycin-resistant Erwinia present in them. And because of that, we are going to have to start making new recommendations for controlling the bloom blight phase of fire blight. And this probably means recommending either kasugamycin or oxytetracycline. So this concludes our lecture on fire blight disease management, as well as all of our lectures on fire blight. I recommend that you review the readings that were suggested, also review the lectures, and at this time it would be good to prepare the study guide for fire blight.